Thank you for attending the Anatomy Department's Centennial celebrations. Both to the people present here and to the audiences joining us virtually, I am Sarah Keshkuli.
And I'm Jay Polepalli.
Please put your hands together to welcome our esteemed guests of honor and most distinguished guests.
I now invite Professor Ong Wei to deliver the welcome speech for the Centennial celebration. Prof. Ong.
Good afternoon, Professor Tan Eng Chai, President, National University of Singapore, Professor Ho Teck Hua, Senior Deputy President and Provost, NUS, Professor Yeo Kay Guan, Chief Executive, National University Health System, Professor John Yuli Wong, NUS Senior Vice President, Associate Professor Benjamin Ong, NUS Senior Vice President, Professor Chong Yap Sing, Dean of the Yong Loo Lin School of Medicine, Professor Barry Halliwell, Senior Advisor, Office of the Senior Deputy President and Provost, NUS, delegates, esteemed colleagues, students, ladies and gentlemen. Thank you to each and every one of you for being with us here today. On behalf of the Department of Anatomy, Yong Loo Lin School of Medicine, NUS, I'm very pleased to welcome every one of you to our department's centenary celebration in the 6th International Anatomical Sciences and Cell Biology Conference, IASCBC. This year marks the 100th year of the founding of the Department of Anatomy. We are proud to gather here today to mark the occasion of the centenary of our department from 1922 to 2022. Our department's strength is in its staff members, and we here celebrate the achievements and honor our emeritus professors and honorary fellows who have served the department and inspired and taught many generations of medical, dental, and life science students with dedication, passion, and excellence. As part of the centenary celebrations, the Department of Anatomy is launching a book entitled Anatomy 100, documenting our journey from 1922 to today. This book is a showcase of our heritage and achievements with historical pictures. We're also launching the Anatomy Centennial Medical Bursary to help needy medical students. In addition, today is the opening day of the 6th International Anatomical Sciences and Cell Biology Conference, IASCBC, where anatomists and cell biologists share their knowledge and experience in both teaching and research in all aspects of anatomy and cell biology. IASCBC was first hosted in 2010 in Singapore, followed by Thailand in 2012, Taiwan in 2014, Hong Kong in 2016, and Malaysia in 2018. As part of the conference this morning, we have successfully conducted the Microscopy Workshop, sponsored by the Microscopy Society Singapore, showcasing the different innovations in microscopy and how these could be leveraged upon to advance our knowledge of microscopic anatomy, really leveraging upon the strengths of the whole entire campus. Best practices in publishing medical education research, sponsored by the Anatomical Sciences Education Journal, as well as the American Association of Anatomy. Over the next few months, you will all be hearing about the department's new initiatives and educational achievements and activities as part of our centennial celebrations. I'm excited to extend a very warm welcome to each and every one of you gathered here today to grace this event. Thank you all for your effort and time. Thank you very much.
My name is S.T. Dean, current head of the Department of Anatomy. The Department of Anatomy is one of the oldest departments in the Yong Loo Lin School of Medicine and is committed to be an active partner in the NUS Medicine's vision that is inspiring health for all. Our vision is to be a leading department spearheading medical education in Asia. The College of Medicine was started in 1905, but the first chair for the department was appointed in 1922. I am proud to mention that the department celebrates its centenary
Congratulations to the Anatomy Department for reaching 100 years of age. The department has a very rich heritage and history and is an important cornerstone of NUS medicine. This is obviously a very significant milestone worth celebrating as we look back at how far the department has come and the many exciting opportunities ahead of it. I wish the Anatomy Department many more centennials as it continues to build on the very good work it has done in educating and inspiring future-ready healthcare professionals. Thank you for all you have done for the school.
One of the most significant things that has happened over the past 10 years is the establishment of the body donation program, whereby Singaporeans could actually pledge their bodies to donate them to the department for the purpose of medical education. And in addition, the department has recently purchased an anatomical table, which allows students to do virtual dissections of the human body. Students can learn in a hands-on way what anatomy is all about. Hopefully this will translate into their performance as better doctors for improving healthcare of our patients.
Recently, a multiphoton microscopy facility has been added. For the last 3 to 4 decades, we have modernized our research and teaching facility in the department.
For the two-photon microscope, there's an option of doing exactly what the confocal does, but more than that, it can image living tissues. This technique has the potential of being able to be used in translational research as well, with the visualization of specific biomolecules in the tissue so as to link structure and function. And that would be very beneficial for medical teaching.
I'm quite inspired by my colleagues in the Department of Anatomy, and to see their passion for education and see how much effort they put into preparation of the teaching materials, and then how they actively approach students to understand what knowledge they want to learn.
Anatomy is a discipline that is actually very intense in detail, and these are all physical details that have to do with structure, but which are important for clinical practice. We actually generated flash files. You're actually building up the structure physically. You could actually click onto the individual piece and try to connect it with the right area in the body. It actually sort of strengthens your understanding of how complex anatomy comes together. The program that the school is doing with Microsoft is ideal for a virtual reality program. So the student could actually come and look at something in the museum, acquire the appropriate images to help understand what they're looking at. So I think that would be a major complement, to the way they're learning now.
Our primary mission is to educate, train medical professional, dental professional, as well as pharmacists. So I can boast today that we have trained many thousands of doctors in Singapore. Anatomy, to me, is geography to medicine. Any medical student, without going through anatomy, is not medicine.
So the Department of Anatomy has a rich history, and we will continue to do our best to train future healthcare professionals for the nation.
Our department has come a long way from its establishment in 1922. We have been constantly reinventing ourselves, adapting to the changing world, and training top-notch medical professionals over these past 100 years. And now, I invite to the dais the head of the Department of Anatomy, Professor S.T. Dean, to deliver the opening address. Professor Dean.
Good afternoon, guest of honor, Professor Tan Eng Chai, President, National University of Singapore, Professor Ho Teck Wah, Provost of National University of Singapore, Professor Yeo Kee Guan, Chief Executive of National University Health Systems, Professor John Wong, NUS Senior Vice President, Associate Professor Benjamin Ong, NUS Senior Vice President, Professor Chong Epseng, Dean, Young Lulin School of Medicine, Professor Barry Halliwell, Senior Advisor, Office of the Senior Deputy President and Provost in US, colleagues, friends, ladies and gentlemen. The Chinese philosopher Confucius is reputed to have said, If you think in terms of a year, plant a seed. If in terms of 10 years, plant trees. If in terms of 100 years, teach the people. This year, the Department of Anatomy marks 100 years of excellence in medical education. Our history goes back to 1922, 17 years after the establishment of our medical school. It is one that all of us associated with the department look back on with quite pride. On this very special occasion, I'm privileged to represent past and present anatomists, researchers, non-academic staff members, and students to reflect briefly on the department's progress. In the early years of medical education in Singapore, anatomy was taught by government medical officers until the establishment of a chair for anatomy in 1922. Professor Gordon Harrower served as the first chair of anatomy at the King Edward VII College of Medicine, Singapore, and rendered great services to medical education in the country for 14 years. The department's proud legacy as one of the academic departments for teaching excellence was built by predecessors. Over the years, we strove hard to build on the legacy of the pioneers with the Singaporeans, Professors R. Kanagosundaram, Wang Wai Chow, Leong Seng Kee, Ling Eng Ang, and my predecessor, Bae Boon Huat, at the helm. Historically, the department has been well recognized for its excellence in teaching human anatomy and innovative teaching methodologies, as well as for its outstanding and dedicated teachers. Many of us still remember Prof. R. Kanagosundaram, fondly known as Prof. Kana. He built both the teaching and research facilities in the department by establishing animal house, animal surgery suite, and electron microscopy facility for research, and human anatomy museum for teaching during his term of head of anatomy. Building on that legacy, we progressively modernized the department's research and teaching infrastructure over time. A cutting-edge molecular biology, tissue culture, electrophysiology, and histology labs, along with the state-of-the-art high-resolution imaging facilities, such as confocal and multiphoton microscopy system, were established to match those in our aspirant departments. Our anatomy museum standing as first-rate anatomy resource center, along with the body donation program, instituted to ensure the sustainability of cadaver-based anatomy teaching, have underlined our standing at the forefront of anatomy education. We are amongst the select few institutions around the world that meld established method of traditional teaching using cadaveric specimens with digital technologies. Our students receive hands-on instruction, working with the cadaveric samples as well as with the virtual dissection table, virtual reality simulation tools, and digital specimens. We have also gone from strength to strength in our health and biomedical science research program, particularly in the key thrust areas of neuroscience and cancer research. These initiatives are reinforced by young and senior scientists who are affiliated to the translational research program focusing on aging and cancer. I'm glad to reiterate that we teach professionally well. Our teachers have been recognized for excellence in this area, with 5 of our senior staff having been designated emeritus professors. Today, we also announced the inception of the Anatomy Centennial Medical Bursary, which will benefit needy medical students who struggle to traverse the medical school due to financial constraints. In conjunction with our Centennial celebration, we are also organizing the 6th International Anatomical Sciences and Cell Biology Conference. This 3-day event features lectures by leading medical educators, anatomists, and researchers working on aging, neuroscience, cell biology, and developmental biology. I hope that this conference will encourage scientific discussions and promote networking among scientists interested in a wide range of basic and clinical aspects of anatomy and cell biology research. Let's begin the Centennial celebrations. I would like to thank President, NUS, speakers, delegates, conference participants, and our guests for honoring us with their presence. I also would like to thank our donors, in particular the members of the NUS Staff Club, for their magnanimous contributions towards the Anatomy Centennial Medical Bursary. Thank you all.
A very good afternoon to Professor Tan Eng Chai, President, National University of Singapore, Professor Ho Teck Hwa, Senior Deputy President and Provost, NUS, Professor Yeo Kay Guan, Chief Executive, NUHS, Senior Vice President, Health Affairs, NUS, Professor John Wong, Senior Vice President, Health Innovation and Translation, NUS, Senior Advisor to NUHS, Associate Professor Benjamin Ong, Senior Vice President, Health Education and Resources, NUS, Associate Professor S.T. Dean, Head Department of Anatomy, NUS, Associate Professor George Yip, Deputy Head, Department of Anatomy, NUS, esteemed colleagues, ladies and gentlemen. To make it to 100 years of age is a truly remarkable feat. For humans, it is very rare, and much research has been done to uncover the secrets of becoming a centenarian. In some ways, I see parallels between the practices and history of the Department of Anatomy and the habits of centenarians worldwide. One of the secrets to living to 100 years of age is knowing one's sense of purpose. The Okinawans, who are renowned for their longevity, call it ikigai, which translates to a reason for being, or more simply, why I wake up in the morning. Since the formal establishment of the department in 1922, the department has not wavered from their key mission of educating and equipping medical students with one of the most fundamental areas of knowledge required of doctors, that of human anatomy. The department has shown us that it is possible to maintain steadfastly the fidelity of their mission and traditions over a century. NUS Medicine, being one of the few medical schools left in the world that still conducts dissections of human cadavers. And at the same time, the department has not shied away from adopting the latest technologies to enhance our students' learning, such as using ultrasound and three-dimensional holographic technology to teach the secrets of anatomy. I'm confident that the department will continue to thrive for many more centuries if they continue to focus on what they do best, which is the teaching and training of medical, dental, life sciences, and other healthcare professionals. Longevity research has also revealed that the world's longest-lived people choose or were born into social circles that supported healthy behaviors. The Okinawans created moais, groups of friends that are committed to look after one another. When practical anatomy was first taught by Dr. G.D. Freer, the first principal of the school in 1905, and his colleagues, and later, when the first chair of anatomy was created in 1922, Professor Gordon Harrower, they brought together a group of outstanding faculty and staff who dedicated their lives to the teaching of human anatomy. When you joined the department, you became a part of this special group, united with a common purpose. And this tradition of commitment and dedication carried on through legendary teachers like Professor A.R. Ellis, Professor R. Kana Gasuntaram, and many, many more, too many to mention, many of whom are present in this auditorium today. Names that will remain names to the younger generation, but whose mention cannot but inspire respect and admiration among those of us fortunate enough to have been taught by them. The Department of Anatomy is what it is today because your predecessors trusted one another and supported each other in pursuit of a shared goal. I hope that you will continue building each other up and helping one another in the years to come. Lastly, centenarians were found to have a positive and optimistic attitude. With the constantly evolving COVID pandemic, we are all uncertain about what the future holds. However, the future is always going to be uncertain, whether we are in a pandemic or not. What we can do to move forward is to set realistic but ambitious and optimistic goals. Start by defining what the department's vision of success looks like, one step at a time. What do you want to achieve by your 110th anniversary? And slowly work your way up to your 200th anniversary. Believe that you will get there and accept that getting there will require focus, commitment, and teamwork. The speed at which the world is changing is scary and increasing all the time. And while I know the department can adapt quickly, take care to forge ahead for a renewed sense of purpose, the same steely sense of purpose that has carried the department through this first 100 years. I look forward to continuing to see great things from the NUS Department of Anatomy. Make the next 100 years another century that future generations of anatomy educators and medical students will commemorate with pride and joy. Happy 100th anniversary, and thank you for all you have done so far.
Thank you, Professor Chong Yap Sing. Please join me as we welcome our next speaker and our guest of honor, President of the National University of Singapore, Professor Tan Eng Chai.
Distinguished guests, colleagues, anatomists, and fellow members of the education and scientific community. Good afternoon to everyone joining us on this day. In the current landscape, being present has taken new meaning. Nevertheless, even as we are drawn in mass and are not able to mingle freely, it is a privilege to gather with each other. My thanks to you who are present, and also to those of you who are with us virtually, especially from all parts of the world. We are most delighted to celebrate a major milestone of the Department of Anatomy at the NUS Yong Lu Lin School of Medicine. It's 100 years of dedication and contribution to medical education. Since the days of its first chair, Professor J.G. Harrower, the department has been a cornerstone of every medical student's education. It is a key, essential discipline that all medical students must master, because understanding the intricacies of the human body is a prerequisite for clinical training. The Department of Anatomy's strength has always been its timely amalgamation of traditional teaching methods, along with incorporating modern infrastructure that technology has made available. Generations of clinicians and healthcare professionals in Singapore have benefited from the learning anatomy through the department's effective blend and integration of traditional and modern technologies. Through the years, teaching has moved from chalkboards to transparencies and overhead projectors to multimedia presentations with the advent of computers in classrooms. In the last 2 years, lessons through online platforms were introduced to minimize disruptions wrought by the pandemic. In keeping with the times, the Department of Anatomy has also embarked on digital transformation with the incorporation of a virtual anatomy museum, virtual dissection table, digitized specimens tagged with QR codes, and telehistology lessons with direct microscope feeds by educators, students, and even prospects of virtual reality learning tools. Now, these technological enhancements have placed the department at the forefront of anatomy education, augmenting its heritage of quality teaching with current as well as next-generation technologies. While technology offers many educational benefits, practical, hands-on learning remains critical. Through what the department calls the Silent Mentor program, medical students as well as surgical residents also gain intimate knowledge and understanding of body systems through cadaveric dissection and the study of prosected specimens. As a testament to the department's quest for academic excellence, the Department of Anatomy has simultaneously maintained a robust research profile. Commencing from the inaugural anatomy chair, Professor J.G. Harrower's publications on anatomical variations of the skulls of different populations nearly 100 years ago. The department has built a rich legacy of research that continues to this day. Broadened by developing technological tools in various facets of experimental research, vis-a-vis modern molecular biology and microscopy techniques, the department's current research profile is in the areas of neuroscience and cancer biology, with an emphasis on translational research that ultimately may benefit society as a whole. The Department of Anatomy will continue its unwavering focus on leading physicians with a good foundational understanding of the human body, nurturing medical professionals who adapt to constantly evolving environments in medicine and medical technology, enhancing scientific understanding through seminal research that explores concepts from underlying molecules to clinical outcomes, and inculcating the drive to be pioneers amongst peers. As we mark the centenary of the department, we also look forward to the next century. The Department of Anatomy has developed distinctive strengths and is well positioned to scale new heights in academic excellence and to inspire generations of students, teachers, and researchers. The 6th International Anatomical Sciences and Cell Biology Conference is being inaugurated today by the department in conjunction with its centenary celebration. Now, on this joyous occasion, I would like to congratulate the Department of Anatomy on its fine achievements and well-earned reputation. And I wish the department every success. Thank you.
Thank you, Professor Tan-Ning Chai. If I may request that you kindly remain on stage, where you will be joined by Professor Ho Teck Hwa, Senior Deputy President and Provost, and the Tan Chin Tuan Centennial Professor. as well as Professor Chong Yap Sing, Lien Yin Chow Professor in Medicine and the Dean of the Yong Lu Lin School of Medicine, and Associate Professor S.T. Dean, Head of the Department of Anatomy. The time has now come to unveil the Department of Anatomy's Centennial book. May I request the President of NUS, Professor Tan Eng Chai, to kindly unveil the book. This book features accounts from some of our dear colleagues, as well as pictures captured through the last century. Can I request Professor S.T. Dean to hand a copy of the Centennial book to our distinguished guests? Thank you very much.
Indeed, it's a journey through time. Now, on behalf of the Department of Anatomy, Sarah and I would like to invite Professor Lee Kim Seng to present a check of $50,000 from the NUS Staff Club to the Anatomy Centennial Medical Bursary. We also request the Dean of the Yong Lu Lin School of Medicine, Professor Chong Yap Sing, to receive this check from Professor Lee Kim Seng. Thank you, Professors Lee Kim Seng and Professor Chong Yap Sing.
May we now invite on stage Professor John Wong, Isabel Chan Professor in Medical Sciences, Senior Vice President of Health Innovation and Translation at the National University of Singapore, and Senior Advisor at the National University Health Systems, to present the Special Recognition Awards. The Department of Anatomy presents these Special Recognition Awards to esteemed individuals in acknowledgement of their valued and steadfast contributions to the department and for their long and dedicated service. To begin with, can we invite Emeritus Professor Rajendran K. Next, we have Emeritus Professor Charanjit Kaur.
Thank you, Professor John Wong. We now invite Professor Benjamin Ong, who is the Senior Vice President of Health Education and Resources, to present the Special Recognition Awards to Professor Samuel Tay. And to Professor Sim Teo Swan. The Department of Anatomy would like to acknowledge Associate Professor Sim Teo Swan for her valued guidance, for going the extra mile when she was the chair of the NUS Medicine Safety Committee in elevating the department's safety and health management. Thank you, Professor Benjamin Ong. And accepting their awards in absentia are Emeritus Professor Ling Eng Ang, Professor Wong Wai Cho, Associate Professor Francis Voon, Professor Leong Seng Ki, Emeritus Professor Gopala Krishna Kon, and Dr. Tan Choon Kim. Dr. Tan Choon Kim is a former staff member, and presently, he's the chairperson of the Kwan In Thong Hood Cho Temple. And this award is for his unwavering and staunch support for of the department over the last 2 decades. And lastly, to Professor Paulina for his valued contributions to anatomy education as a consultant in our department, for the education program, and for his strong support of anatomy education-related activities in the department for the past 15 years. We will make sure these awards get to them safely in a timely manner. Once again, I request you all to put your hands together to applaud all the awardees of the Special Recognition Awards. Thank you. We stand on the shoulders of giants. As the anatomy department completes its first century, let us hear about the journey of some of the giants who have contributed and are still contributing to the department's legacy. This legacy that we will carry forward into the next century.
I'm Prof. Samante.
Hi, I'm Chiranjit Kaur.
So I am Professor Gopal Krishna Kon.
I'm Bibun Huat. I'm Associate Professor Dinesh Kumar Srinivasan.
My name is Song Lin.
I am Sarah Kashkuli.
Hi, I'm Guo Xiang.
Hi, my name is Safira.
The teaching load, you can say it was probably close to 300 hours a year. So, but of course, with the help of excellent colleagues, both academic and non-academic, I was able to sail through.
I got an opportunity to work in this department as a teaching assistant in 2003. Since then, I've been teaching anatomy.
And I'm on the educator track, educating our medical, dental, and life sciences students, just teaching them anatomy and embryology.
So I actually started off as an admin. So later on, I heard that one of our colleagues was retiring, and actually I've been very interested in the body donation program. I decided to take up his position.
I first joined in February 2017 as a research assistant under Prof. Bay. And then in 2018, I transitioned to a lab tech.
Initially, I was here just to do animation, working closely with Prof. Raj.
I organized many international conferences. I also invited very few ministers and also a lot of WHO workshops.
I first joined and having to do full-time dissections with students. In '86, I have to grapple with all the knowledge I needed to start teaching, so it was quite a mammoth task for me. Yeah.
So I still remember that time when I shared a room with Prof. Day in Japan, and on the second day, in the morning, he suddenly woke me up and saying that we are really late because it's late morning, and I jumped out of bed only to look at the clock and found that it was 4:30 a.m.
I remember people better. Working with people. Those are the memories that I treasure more.
The department has done very well, and it is also commendable that in the latest QS World Ranking, the Department of Anatomy and Physiology were ranked 21st in the world. So to me, this is no mean feat.
I'm glad that we have quite diligently documented many critical junctures of the department's journey.
Very proud that I have contributed and am part of this excellent department.
I feel very honored because 100 years of history with the School of Medicine, so yeah.
From the time we learned anatomy and now it has changed a lot.
It is quite an honor to be able to walk where giants have walked. We've had so many great educators that have been a part of this department, and it's an absolute honor to now have a little part to play in contributing to that too.
So with the help of technology, and of course with the retaining specimens that we have, I hope that students will become great doctors who will continue to help people who are in it.
It was quite an inspiring thing for me to relook at the past and to encapsulate what the department has done in our own 100 years. And I think we have done a lot, we have progressed a lot.
My wishes and hopes, they are that the department should continue to excel as it has done in the past 100 years with dedicated staff in teaching, in research, in administration, and whatever else the department needs.
I hope the department producing generation of doctors who are in synchronized with medical technological advances.
Well, the last 2 years have really accelerated our digital growth, and I hope that we continue on that same trajectory so that we can reach our audiences, reach our students, not just locally, but globally as well, and try to get anatomy education out to as many students as possible and continue on our path of academic excellence.
We had to devise a different way of learning and teaching anatomy. So digital technology is the key, and anatomy is the pillar, foundation.
I still hope that our department will continue to strive to help out many other people and continue to train great professionals in this field.
The department will be more well known to even people who do not study here, because ultimately schools are meant to reach out to people like us also.
Medical education is continuous, and the students need to have good foundation in gross anatomy as well as histology to perform well in their career when they graduate. And I believe that anatomy in the next 10 years and more will be the basis for clinical practice. Yeah.
So for the next 100 years, I would like to wish the department a smooth sailing journey as it forges new and exciting horizons, and I hope that you will achieve even greater heights. So long live the Department of Anatomy at NUS Medicine.
Thank you for joining us in today's very special Centennial celebrations. We would like to continue this afternoon by inviting Professor Yeo Kay Guan, Irene Tan Liang King Professor in Medicine and Oncology, Senior Vice President of Health Affairs at the National University of Singapore, and Chief Executive of the National University Health Systems, as well as Professor S.T. Dean, Head of the Department of Anatomy, If you could put both, please ring to start the 6th International Anatomical Sciences and Cell Biology Conference. Thank you very much, Professor Yoke Guan and Professor S.T. Dean, for ringing in the start of the 6th IASCBC. Please now join us in welcoming Associate Professor George Yip, the Deputy Head of the Department of Anatomy, to share with us his vote of thanks.
A lot of work has been put into the Centennial celebration of the Department of Anatomy and the 6th International Anatomical Sciences and Cell Biology Conference. The organizing committee would like to present the following vote of thanks. First of all, we would like to thank our guest of honor, Professor Tan Chai, and our distinguished guests, Professor Ho Teck Hwa, Professor Yoke Guan, Professor Chong Yap Sing, Professor John Wong, Professor Benjamin Ong, and Professor Barry Halliwell, for taking time off their very busy schedule to attend this afternoon's event. We are indeed honored to have you with us. The success of the conference depends on the sharing of scientific ideas and discoveries. We would like to thank our keynote speaker, Professor Brian Kennedy, and all the invited speakers and oral and poster presenters for sharing the fruits of their research with us. We would also like to thank the chairs of the scientific sessions, the conveners of the workshops, the judges, and the oral and poster presentations, as well as all the delegates and participants. Additionally, we would like to thank our sponsors for their generous support. They are the Anatomical Sciences Education Journal, which is the official publication of the American Association for Anatomy, Geo Asia Private Limited, Leica Microsystems Southeast Asia Private Limited, Microscopy Society of Singapore, and 10x Genomics. The Department of Anatomy recognizes that financial support is important for the education of our needy students. We would like to thank the NUS Staff Club for their donation to set up the NUS Staff Club Anatomy Centennial Medical Bursary. We also thank the kind sponsors and donors who have contributed to the Anatomy Centennial Medical Bursary. Much effort has been put in over the past 3 years to ensure the success of this large-scale international 3-day events. We thank everyone in the organizing committee, as well as all the staff, students, and vendors who have contributed their time and energy over this long period. Finally, and importantly, we thank all the past and present academic and non-academic staff and students of the Department of Anatomy for their dedication, passion, and hard work over the past 100 years. Their contributions have led the department to its current global status, where it is ranked 21st in the world, together with the Department of Physiology. Thank you very, very much.
Thanks, Professor George Yip. As we make our way to the keynote lecture of the 6th IASCBC, how about we put our hands together for all the esteemed guests as they take their leave.
The topic of the first plenary lecture for the 6th IASCBC conference is Addressing Human Aging, No Time Like the Present. This topic is important and of great interest to us because aging research has slowly gained momentum over the last 3 decades and has now become a mature field of scientific endeavor. Given that over 20% of the population will be over 65 years of age in a not-so-distant future, it is imperative that strategies are developed to slow or reverse the aging process. Our speaker for this talk is Professor Brian Kennedy. Prof. Kennedy is internationally recognized for his research in the basic biology of aging. He is a distinguished professor of biochemistry and physiology at the Yong Loo School of Medicine at the National University of Singapore. And he also serves as the director of the Center for Healthy Longevity at the National University Health System, as well as the director of the Healthy Longevity Translational Research Program at the Yong Loo School of Medicine and the Asian Center for Reproductive Longevity and Equality. Professor Brian Kennedy was also the director of the Buck Institute of Aging in USA. Professor Kennedy has been involved in several technology companies focusing on aging and age-related disease. Ladies and gentlemen, let us welcome Professor Brian Kennedy.
Thank you, Dr. Ong, for the introduction, and Professor Dean and the organizers for inviting me. It's a great honor to give this lecture. I want to also thank the distinguished guests, Ing Chai, Tekwa, John, Kegwan, Barry, all had a major role in either inviting me here or bringing me to the, setting up my lab for the Singapore. And most of all, I want to thank Yap Singh, who had the foresight to email me the night Donald Trump got elected and said, "You need a new country." And he's been a strong advocate of the research here. I've definitely benefited greatly from coming to NUS and setting up a lab, and I'm very happy to be here. I want to just add one thing to Confucius. He said, I believe what I heard is if you want to think about 1 year, you should plant seeds. 10 years, you should plant a tree. 100 years, you should educate the public. And I think what he left out was if you want to think 150 years, you need longevity medicine. So I'm going to try to tell you today a little bit about the development of drugs and other methods to slow aging. I'm not going to spend too much time. See if this works. I'm not going to spend too much time talking about introduction here. I've given this introduction many times. But I do want to spend just a little bit of time emphasizing the importance of aging, and then I'll get into the science. I think right now, still, we mostly think about research and medicine in terms of boxes. So you have all of these chronic conditions that we spend a lot of research dollars on, that we develop specific therapies for, that you have specific doctors who are specialized to treat. And what we've ignored is the fact, largely, that aging is the biggest risk factor for all of these chronic diseases. By far the biggest risk factor. And that leaves a relatively simple thought that if we could target the aging process and move from sick care to health care, we can prevent many of these chronic conditions simultaneously. And that's really the goal of my research and most of the aging research field. Now, many of you may be distracted, I know everyone has been in the last couple of years, by the COVID-19 pandemic, but it really doesn't change the equation at all. The biggest risk factor for mortality due to COVID-19 is being over 70 years of age, and that's independent of other other risk factors. But that's not even surprising because it's also the biggest risk factor for mortality due to influenza and many other infectious diseases. So really, when we have this slide that aging is driving disease, we should be mentioning it this way, that aging not only drives chronic diseases, it drives the ability to respond to acute infection, and it drives functional decline. So maybe we should think about targeting this process as getting the biggest bang for our buck in terms of extending health span in Singapore and globally. And really, this is the crux of the research that we're trying to do. If you imagine your life as a timeline, a person is born, they're healthy for a long period of time, they start getting chronic diseases, they get multimorbidity, and eventually they succumb. Right now, we're spending almost all of our medical research, not research dollars, but also treatment dollars in this red phase, when people already have chronic diseases, and in most cases, we don't cure them completely. We help them manage these diseases. Now, there's nothing wrong with that. If I get sick, I want my diseases managed too. But I think we're forgetting the green area. And that if we can do interventions earlier while people are still healthy, we can be successful at keeping them healthy for a longer period of time, compressing morbidity. We'll extend lifespan, but the main point is we'll extend health span greatly. And that's really the goal of aging research, true health care. Now, you might think that it's not possible to do anything about aging, but we've learned a lot about the mechanisms that drive the aging process over the last 30 years or so since I've started in the field. It's funny, when I started, people would say that in 1990, people would say, well, by 2020, surely we'll know everything there is to know about aging. We probably won't be able to develop interventions because it's such a complicated process, but we'll learn a lot. And while we have learned a lot, I would argue we still don't really know what causes aging. We're still debating about that. But it's been the total opposite. Developing interventions has been relatively easy, at least in animal models. And so the question that we need to ask, and the main purpose of this talk, is can we develop these interventions, understand what they do, and implement them in human populations? So there's a lot of names on, these are two reviews that were written in the 2013 and 2014 that have been popular in the aging field, and they identify pathways that drive aging. Hallmarks on the left, pillars on the right, and you can read them. I don't think it's important to go through these in detail for today's lecture. The point I do want to make, though, is that these are just readouts of aging. I think what's truly happening here is you have a homeostatic network in your body that works to keep you healthy. It's not there to make you live a long time. It's there to keep you healthy and improve your fitness so you can reproduce, generate the next generation, raise kids, and stay healthy as long as possible. But when enough damage happens, this network starts to break down. And when it breaks down, things spin out of equilibrium, and that's when you start getting chronic diseases. So aging interventions are not necessarily about fortifying one of these pillars. What they are probably doing is hitting nodes in this network that preserve network function, and you can read that out by improvements in all of the pillars. And so we're really talking about systems biology. And actually, interventions have been relatively easy to come by, at least in animal models. There are three main types of interventions. There are lifestyle interventions, which are in this dark red circle, and those are highly researched. They're fully researched here is an overstatement. That's from a review. But they're highly researched, and we know largely what to tell people to do to stay healthy. The challenge is to get them to change their lifestyle in a way that will actually keep them healthy longer. There's an intermediate circle here, which is drugs, small molecules, and that's where I'm going to focus my attention today. And then there's a kind of a radical futuristic approach, stem cells, regenerative medicine, gene therapy. These are things that are not ready for prime time yet, but they could have even bigger effects on lifespan and health span than the drugs and lifestyle changes at the end of the day. So I think it's important that we think about the future, but where our focus right now is testing how to do things that will extend human health span today. Now, as I said, there's no shortage of potential interventions in model organisms. The ones on the left are ones I work on in my lab. I'm going to tell you a little bit about alpha-ketoglutarate and gemfibrozil today, two very different kinds of molecules. But this is the drug age database, and there are now over 1,000 distinct drugs that have been reported to extend lifespan in some animal model, and 110 in mice, if you want to think about mammals. And so the challenge is pretty obvious. Which of these 110 interventions will work in humans to extend health span? And what is aging research now? It was basic biology when I started, and there's a lot of mechanistic research going on that's important. There's translational research, and I'll tell you a lot about that today. Clinical research to validate whether interventions affect aging, and really implementation science, which I would put in the category of how do we induce people to develop lifestyle interventions that we know are healthy? How do we change societal structure to create a healthy environment for people? It really crosses a number of boundaries. I'm not going to talk much about this last topic, but I'll focus on the first three today. And I'm going to do that through the eyes of two different molecules. One is alpha-ketoglutarate, which is a natural product. It's a component of the TCA cycle. And another is gemfibrozil, which is in a class of drugs termed fibrates, which I'll come to in a minute. So this would be a off-patent drug that's widely used for hyperlipidemia, and it's quite a cheap drug now to use because it's off-patent. So first, the AKG. This is a collaboration with a company, Ponce de Leon Health, and what they were interested in doing is finding combinations of natural products that would have synergistic effects on aging. And so first, with Gordon Lithgow, we did these screens in worms and find combinations of things that extend lifespan even more than either one alone, and then took the hits and moved them into mice, and that's where my lab came into play. And we did interventions in mice at 18 months of age and looked at their natural lifespan. But also we looked at frailty, we looked at biomarkers of aging, and we looked at parameters of aging like immune changes, adult stem cell function. And I'm not going to tell you about the combinations too much today because they're more complicated. I'm going to focus on the main molecule that had the biggest effect, and that's alpha-ketoglutarate. So this is what happened. We gave it to mice at 18 months of age. It extended female lifespan by about 5 to 10%, and that was significant. There was a trend toward lifespan extension in males. We've done this 4 times now to get the same result. The point I'll make here is that this is a modest effect in a mouse. You wouldn't get extremely excited about this result. It's interesting, but there are things like rapamycin that have a much bigger effect. The exciting thing about alpha-ketoglutarate is what it did to health span. And this is probably the most complicated slide I'm going to show you today, so bear with me. What we're looking at here is measures of frailty in individual animals, each circle or dot. And the higher the score, the more frail the animal is, and we're looking starting at 18 months out through the natural lifespan of the mice. And what you can see is the control animals get more frail more rapidly than the alpha-ketoglutarate treated animals. But this is a complicated way of looking at this data because this animal here, for instance, was highly frail at 23 months, and it's not on the curve at 25 months because it's dead. And so we recalculated this after all the animals were dead by the percentage of the lifespan the animal was at when we did the frailty measure. And what you can see is frailty goes up quite linearly in the control mice, but it's largely suppressed in the alpha-ketoglutarate treated animals. So what we're doing here, and this is true in females and males, so what we're doing here is we're only extending lifespan by 5 to 10%, but we're extending health span by 50%, and that's compressing morbidity. So if you want to think about an economically viable approach to making humans healthy longer, the best thing you can think about is keeping them healthy longer and compressing morbidity. And at least in animals, that's what this natural product does. What is alpha-ketoglutarate? So as I said, it's a TCA cycle component. It's involved in energy production. It's involved in amino acid synthesis and breakdown. It's a cofactor for about 500 different reactions in the cell, which makes it really hard to figure out mechanistically what it's doing. And I'm not going to spend time talking about that today, although we have some ideas. We've shown that it improves adult stem cell function and improves metabolic flexibility, enhances mitochondrial respiration, interesting changes to the microbiome that we're fascinated by. I'll mention the inflammation and the biomarkers. So this is the inflammation data. What you're looking at here is we've normalized all these inflammatory cytokines to 1, or this orange color here on the bottom. In the second row, you see 28-month-old control animals, and the more red it is, the more of these inflammatory factors they have. And you can see that when you treat these animals with alpha-ketoglutarate, you largely suppress that increase in inflammation with aging. And we know that inflammation is one of the big drivers of aging, so this is a very encouraging piece of data. I'm not going to show you the details on the bottom. But that's mouse data, so I wanted to show you some of the latest human data we have on AKG. And what we're using here, and also in the clinical studies in US I'll tell you about later, are biologic aging markers. So we now have biomarkers that can estimate a person's biologic age. And they include things like blood chemistry clocks, and I'll mention one of those in a minute, DNA methylation clocks, the changes in methylation, mostly at CPG islands and genes, transcriptomic clocks, many others that I'm not going to talk about today. These have all been developed in the last 10 years, and there's still questions around accuracy, reproducibility, applications, et cetera. But I think most people in the field are coming to accept these biomarkers, and what we want to do is use them as endpoints to see if our aging interventions are effective or not. So in other words, if your chronologic age goes up linearly, your biologic age may be quite different than that. And so you may be not aging well, so you're biologically older, or you may be aging very well and you're biologically younger. We can measure that now, and I'm going to show you some of the data. So the first study I'm going to show you is people using the Rejuvent product. And so just to be clear, this is alpha-ketoglutarate plus low-dose vitamin A for men and plus low-dose vitamin D for women. And we had 42 participants that were buying the product and willing to give us their data. And they did DNA methylation testing for their biologic age at the start and an average of 7 months after taking the product. These were people an average of about 65 years old. And what we found here on the left is that they get biologically younger, at least by this methylation clock. Now, this is not a placebo-controlled study. It has many limitations, but it's the first piece of data we could get. It's also a relatively simple methylation clock, and there were reasons we chose to use that for this study. What you're looking at here is their baseline biologic age relative to their chronologic age. And these are people that are younger. These are people that are biologically older. And you can see that biologic age gets younger for almost most of the people taking the product in this alpha-ketoglutarate study, or Rejuvent study. Now, interestingly, what we found is that it doesn't work equally well for everybody. There were two parameters that would predict the level of response you had to this product. One was your chronologic age, and the higher your chronologic age, the more response you had. And the other was the differential between biologic age and chronologic age. So if you're biologically old relative to your chronologic age, you respond better to this product. This is not exactly what we were expecting. I don't know if it's going to be true for all aging interventions or just this one, but it's a fascinating piece of data, and this was just published. We've been able to go back now and do a placebo-controlled study, and I'm just going to show you one piece of data from this because we're still analyzing the data. I should say that Diogo Berardo, a postdoc in my lab, is doing, the studies are being done in the US, but Diogo is doing all of the analysis, the statistical studies. So this one, we enrolled 100 subjects at Indiana University. 78 completed the 9-month intervention. There's a placebo group and an intervention group that's split between males and females. Average age is between in the mid to late 50s. The span is between 46 and 66. And we chose that age range specifically because we think those are people that are probably not yet have many chronic diseases, but they're going to get them soon, so they're susceptible, and that we think that's the first place to do longevity intervention studies. And this time I'm going to show you a different clock. This is Morgan Levine's phenoage clock. So unlike DNA methylation, this is developed by metabolic markers in the blood that you're all familiar with. And if you weight them appropriately, you can predict biologic age with more or less the same accuracy as the methylation clock. And so when we look at this clock, we find very similar results. So there's no effect on the placebo group. They got 1.1 years older in the 9 months of the study. But the Rejuvent group, by this clock, got 4.2 years younger. So this is a statistically significant change. And again, the main predictor was if you're biologically older than your chronologic age. So people that are not aging well by these clocks are the ones that are responding the best. Now, I have to add that we didn't get a statistically significant signal in the methylation clock in this study, and we're still looking at a number of other clocks. So this is early days, but I think it's very positive news that at least this intervention may actually be affecting human aging. And there's, like I said, this is just one. There are many other interventions, natural products and drugs and otherwise, that are being tested. And we need more studies like this to demonstrate whether they actually work in humans or not. So I'm going to come to gemfibrozil, and this started off a long time ago when I was at the Buck in a collaboration with Michael Palominas at Texas A&M and a postdoc, Chong He, in my lab, who's now doing very well in the biotech industry. And we were able to use yeast cells to find, I'm not going to stay on yeast for just a couple slides, we were able to use yeast cells to find drugs that slowed aging in yeast and then test whether they were conserved in other species. The first one we found was ibuprofen, and we published a paper on that. The second one we found is gemfibrozil, and that's the data I'm going to talk to you about today. Now, when we studied ibuprofen, we were able to show that what it was doing was decreasing the stability of tryptophan transporter in yeast cells, and that was critical for turning down TOR signaling and extending lifespan. And that's not that surprising, because we've known that amino acid restriction has extended lifespan in mice, worms, flies, and yeast for many years now. And so this is a drug that may be mediating the same effects as amino acid restriction. But this was also a clue, because by virtue of how we did the screen, it suggested Gemfibrozil might be working a similar way. Gemfibrozil not only extends lifespan in yeast, but it extends lifespan in worms, so it's conserved at least between those two species. And it extends health span in worms too, so it increases the activity of the worms, it provides resistance to reactive oxygen stress, and many other things. Now, what is Gemfibrozil? So if you're a clinician, you probably know what it is. In the US, it goes by the trade name Lopid. This is a fibrate, or at least it's proposed to be a fibrate. I want to argue that that may not be the case. Here's the structure. It was developed in the 70s as a hypolipidemic drug for management of hyperlipidemia and coronary heart disease. It's usually secondary in use to statins if there's some contraindication there. And this was the data that it lowered triglycerides and increased HDL cholesterol in clinical studies, and there have been many studies since then. So what's the proposed mechanism of action? Well, this is a complicated slide, but the general gist of it is that it's a PPAR alpha activator. So this is a transcription factor that controls a number of metabolic features of lipids, and Gemfibrozil was proposed to bind to PPAR alpha, relieve it from a repressor complex, allow the transcription factor to go in the nucleus and activate transcription. I want to argue that it's not doing this. We have no evidence in our cell culture studies that has this effect. And if you go back and look at the original data, there's only about a twofold increase in activation of PPAR alpha targets, even in the best studies. And I think it was convenient to call this a fibrate like many other fibrates that have this effect. But I don't think that the activity is going through PPAR alpha. And we can show that the lifespan extension in yeast and worms does not require the orthologs of PPAR alpha. So the data I'm going to show you is from Chong and also from a graduate student here at NUS, Charmaine, who's really doing a fantastic job with the research. I'm very lucky to have a number of great graduate students. Charmaine is the most senior, and she's really doing a great job. So if we go back to the yeast, we were able to show that it's about amino acids again. So you can ask yeast cells to grow on 78 or 80 different carbon sources and say, where does this drug cause problems? And this drug didn't affect growth on most of them, but if you ask the cells to grow on dipeptides as a source of energy, Gemfibrozil blocked growth. And so that suggests that it was inhibiting uptake of dipeptides and tripeptides in the cell. And if you look at the amino acid levels, what you find is a relatively significant reduction in levels of amino acids inside the yeast cells. This is one of the kinds of elegant experiments you can do in yeast. You can add a toxic non-native dipeptide that when it gets in, it kills the cells. And so if you don't have the drug, you can see this zone of inhibition. You put the drug on this piece of paper, the yeast cells are growing on the plate, and you can see that anywhere that not the drug, you put the dipeptide. Anywhere the yeast that are exposed to the dipeptide are unable to grow because of the toxicity. And Gemfibrozil blocks that effect. So that's very consistent with it inhibiting dipeptide uptake. Same thing in worms, it inhibits dipeptide uptake in the gut. And the same thing in human cells, we can show a reduction in dipeptide uptake in PC3 cell culture cells. So this seems fairly consistent, and in fact, we can show in human culture cells there's a reduction in amino acid levels as well. So when we got here, we wanted to know what the target of Gemfibrozil is, and it's nice when you do a screen and you get something that should be the target, and that's what happened. This is a collaboration with Par Nordland and his team at IMCB, who've been extremely helpful to what we're doing. And so essentially you expose an extract from cells to a drug, and drugs typically when they bind to a protein in an extract will stabilize it. So then you just raise the temperature and the protein denatures, and you have a way of getting rid of the denatured protein. And you look for proteins that are stabilized in the presence of the drug. And the one we found is a PEPT1, which is a dipeptide transporter. It's not the only one we found. I'll come back to another one in a minute. And you can see the data here. Here's temperature increase in the absence and presence of the drug. You can see that as you get to higher temperatures, the protein becomes destabilized in the absence of the drug, but it's stabilized in the presence of the drug. So this is a nice result. So what is PEP-T1? Belongs to the SLC15 family of transporters. It basically exists in the lumen of the gut and in enterocytes, and it's an active transporter of dipeptides and tripeptides. Now, everybody likes to think of specific amino acid uptake molecules or transporters for individual amino acids like the tryptophan transporter I mentioned. But your gut is a messy place. It doesn't produce a lot of pure, clean amino acids. What you're really taking up are breakdown products from food, which are often dipeptides and tripeptides. So these are, and polypeptides. And so these are significant sources of amino acids. And so amino acids, as I mentioned earlier, are strong activators of the mTOR pathway, and we know that TOR is the most recognized pathway in terms of aging. TOR is activated during the aging process. If you use a drug like rapamycin to turn down TOR, you delay pathology and extend the lifespan of the organism. So it's not surprising that what we're seeing here is something that feeds into mTOR signaling by blocking the role of amino acids and activating mTOR. And this is preliminary data, but I'll show it to you anyway. So if you take cells that are starved in culture, they have very low TOR signaling, and that's measured by phospho S6 kinase, and it's target of the TOR kinase. You can activate that with leucine or with amino acids, and then you get increased TOR signaling. And in those cases, when you use single amino acids, you don't get inhibition of TOR signaling with gemfibrozil. But if you activate it with a dipeptide mix, you get increased TOR signaling, and now you block that with gemfibrozil. So this is a very nice affirmation of the model that we have. Now, so we think that we have a new target for aging, and that's a dipeptide transporter in the gut. We're now showing that when you isolate the gut from a mouse and do amino acid uptake, we can actually block the activity of this transporter. And so we think this is another interesting molecule to test in the context of aging. Now, another target we got out of the screen is FABP4, which is a promoter of atherogenesis and dysregulated lipid metabolism. There's been a lot of research lately on FABP4, and you can see that there's a stabilization of the protein when you add gemfibrozil. And so our working model now is that the target for hyperlipidemia is probably FABP4 and not PPAR alpha. And that may also contribute to the beneficial effects on aging in addition to inhibiting the dipeptide transporter. So we're at an earlier stage here with gemfibrozil. We're just doing the mouse lifespan studies now, but this is another intriguing molecule to look at in the context of human aging. It's a generally relatively safe molecule, safer than other fibrates, by the way, that's widely used and may be slowing aging as well as affecting hyperlipidemia. And I would argue that a lot of these drugs that have been developed for early stage risk factors for diseases, I didn't talk about metformin, but it's a known longevity drug, statins, beta blockers, and many others are probably having some of their effect by affecting the aging process as well as the disease they were developed for. So we should really look at repurposing a lot of these early stage disease drugs to see if they impact the aging process. And so what I'd like to do here is focus on what we're doing in the clinical studies, which are still really being set up, but we're making progress now. So we have a Center for Healthy Longevity at Alexandra Hospital now. You're welcome to come over and see it if you're local. And we're doing active studies on human populations to measure the rate that they're aging and to look at whether interventions affect biomarkers of aging. The first study we're doing is called ABIOS. This is a cross-sectional measure of 450 people across age in the Singapore population and across ethnicity. We're about halfway done with this study. And we're starting intervention studies with alpha-ketoglutarate and hopefully rapamycin in the very near future. So what do we need to do to get human? First of all, we need to get the right people, and Andrea Meyer is definitely the right person. She's a geriatrician, but also a gerontologist and internal medicine expert who's come to Singapore recently to work together with us to try to really move these clinical studies forward. So we need to validate that things work in humans. Ultimately, we need to personalize what interventions we choose for which people. And we're just starting studies in both mice and humans to see if we can do that personalization. And we need to do it in a way that scales. And so what we want to do in US are not test $100,000 stem cell injections that we can't scale, but to test things like supplements, repurposed drugs that may have a very significant effect and that we can rapidly move into the population. That also goes with biomarkers. We can't use muscle biopsies because we can't scale that. But if we're taking things we can measure in blood or saliva or physical function, appearance, to measure biologic aging, those are things that are easily scalable. So we have an eye toward the end product, even as we're getting started. So I mentioned this clinical study that was already done and a little bit of data from that. We're going to be doing similar studies in US as a first line, which is a sustained release calcium alpha-ketoglutarate without the other vitamins, because we want to understand mechanistically what's going on. But otherwise, it's very similar, except that we're going to look at a lot more parameters of aging than was done in the IU study. And so I won't spend too much time on that. I do want to talk a little bit about biomarkers, though. And so now we have a whole range of different kinds of biomarkers to measure aging. What we need to do is to figure out how we can use that to distinguish between biologically young people and biologically older people that are early pre-disease. And we need to see which kinds of interventions have effect on which biomarkers of aging. So our goal is to test 5 to 10 interventions, not 1 or 2, and compare and contrast which ones work best with which biomarkers. And they could be micronutrients, it could be dietary interventions, it could be exercise, and I mentioned already work with repurposed drugs. Now, there are many, many different kinds of biomarkers in the population that have been developed in the last 10 years. Classically, what you used are physical function, like time, get up and go, walking speed, grip strength. Those work fairly well in over 70 population at predicting mortality, but we need something to measure aging across the whole age range. And so there are individual markers that have been used, which have some efficacy, but not that much. What really changed the field is the development of omics-based molecular pattern biomarkers. And I mentioned epigenetic clock already. There's transcriptomics, proteomics, metabolomics, microbiome clocks now. And we're trying to understand how they interact with each other and which ones predict which aspects of aging. So in our studies in US, we're not choosing one. We're going to choose several, including DNA methylation, autoantibody panels to look at systemic inflammation markers, measures of senescent cells in the population. We will look at telomeres. It's not my favorite marker of aging, but we will look at that. And then physical activity function, DEXA scanning, pulse wave velocity. And so we can understand how they relate to each other and try to develop a simplistic biomarker that doesn't require 20 different things to measure a person's age. Also, we're re-evaluated how we do our mouse studies now, so they're more like the human studies. We start in late middle age, treat for 6 months, and look for effects on biologic aging markers and frailty. And by doing that, that aligns how we're doing the human studies, and it'll allow us to work iteratively back and forth to improve how the mouse studies predict human outcomes. We have a lot of mechanistic stuff going on, precision dosing that I don't have time to talk about today. And really, I think this is the crux of the question in aging right now, is that now we know mostly the pathways that are driving aging. We know the biologic aging markers, or at least some of them, and we know the diseases that aging drives and how to measure markers for those diseases. And so we're putting together a team of researchers now at NUS with the goal of connecting these things up. Which of these biomarkers best measure which pathways of aging? And we're going to set up a core to measure all of these pathways of aging that can be used by scientists here at NUS if they're interested. And which of these biologic aging markers predict different diseases? We've only chosen 3 here because of bandwidth issues, but we could choose 20 if we wanted. This is metabolic disease, cancers, and response to viral infection like COVID. And so I think what we can do if we do these studies right, and these are mostly human studies looking at biomarkers, is begin to not just weight all of these lines evenly, but figure out which lines connect most to which pathways and which diseases. And I think if we really want to understand how a person ages and why they get sick, this is how we need to take the approach, not studying one disease at a time, which tells you interesting information, but it tells you in a very limited scope. We need to look more holistically at what's going on. So we're going to start with small studies, go to RCTs, and then hopefully go to community translation as quickly as possible. Things like the health district may enable that, and so we're excited to work with people that are really looking at community population healthcare. And then finally, most of you know, but we've set up a healthy longevity program here in the US. This slide's a little bit old, but we have nearly 40 faculty working on aging or various diseases of aging. And it's an exciting team, and we're still trying to put that together to be most effective at understanding aging and how aging drives different diseases. We've also started an Asian Center for Reproductive Longevity and Equality, well, again, with the help of Yap Singh, where we're trying to understand why ovarian function declines so early in the aging process, what can be done about that, and the links between ovarian aging and systemic aging in the body, which are definitely profound but not well understood at this point. And then finally, if anybody's interested in aging seminars and learning more about the aging process, we have a weekly one at 7 p.m. on Thursday through NUS Medicine. We've had about 35 different speakers on. You can go to a YouTube channel and get all of the talks on specific topics of aging after the fact if you don't want to watch it live. And we have some of these shows have up over 100,000 views now. So this is becoming pretty widely watched. And we're hoping to use this to really disseminate not only to Singapore but globally the revolution that's happening in the aging research field and what that might mean for human health over the next couple decades. So with that, I think I'll stop. I talked a little bit about studies that started at the Buck, but most things are happening, almost everything's happening in NUS now. Jor Ming is working on the clinical studies. Shoping has helped out with AKG. Charmaine is working on Gemfibrozil. Max and Diogo, especially Diogo, have been analyzing data from the human clinical studies with alpha-ketoglutarate, and I'm very fortunate to have a lot of collaborators that have helped out as well. So I'm happy if there's time to take questions, and if not, you know where I live. You can contact me and send me emails. Thanks a lot for your time, and thanks for inviting me to give this lecture.
Thank you, Brian, for the very, very interesting talk. Okay, so on behalf of the IASCBC, we'd like to present you a token of our appreciation.
All right, thank you very much. It's been a delight spending this afternoon with everyone here today. Thank you again to our guest of honor, distinguished guests, and everyone joining us both online and in person. We hope you enjoy the rest of the IASCBC conference. Have a great day, everyone.